start talking about the phases of regulation of the gastrointestinal control, right? There are three phases: the cephalic phase, the gastric phase, and the intestinal phase. The cephalic phase is pretty much originating in the head, so everything above the stomach. The pharynx, the nose, right? All of our senses, thoughts, taste, smell, and I dare say sound, right? The sound of probably ice cream truck or maybe popcorn popping will elicit this response. And these are going to input signals into the long reflex pathway of the central nervous system, then to the enteric nervous system, right? That long axis that we talked about just now. The gastric phase is going to be originating in the stomach, so the presence or absence of food in the stomach, the presence of those molecules, the presence of water in the stomach is going to initiate this response, and that is going to feed into both the long and the short reflex pathways, as well as some hormones that we're going to talk about. And then the intestinal phase, same deal. The presence or absence of these substances within the intestine will be the stimulus for both the long and short reflex pathways, as well as some hormones that we will talk about. All right, so the one that's sort of unique here is the cephalic phase. This is only the long reflex. There's no short reflex involved in the cephalic phase. And that makes sense, right? When you see something, when you elicit those senses, it has to go through the central nervous system. Therefore, it has to be the long reflex pathway. All right, moving on to our second objective, we're going to talk about the function of saliva and stomach acid, as well as pancreatic juices and bile, and how those substances are regulated. We'll talk about salivary secretion, we'll talk about acid secretion, and then we'll finish up by talking about bile and pancreatic juice, and we'll look at motility very briefly. Now, salivary secretion is elicited in response to the taste of food, like the physical presence of food in the oral cavity, as well as the texture of food. So different textures can stimulate different types of mechanoreceptors specifically, whereas the presence of certain molecules in food will elicit a chemoreceptor response. So this all takes place within the oral cavity. This is going to relate to the salivary centers, or the control centers within the medulla and the brainstem, and this is where the autonomic regulation takes place. So from this point, we will signal the parasympathetic nervous system to increase those salivary secretions. Right? So the presence of food sends signal to the central nervous system, specifically the medulla, those control centers. That sends a signal via parasympathetic autonomic nerves to increase salivary secretions. So again, this does not have to be physical. This can be sight, smell, all those different tactile elements which feed into the cerebral cortex and stimulate the salivary centers that way. Now, we know that there are two types of salivary secretions based upon which division of the autonomic nervous system is engaged. So the parasympathetic activation is going to bring about a more watery saliva. So this saliva has enzymes such as salivary amylase, which we talked about, but it has a lot more of a water content. And so it's going to be more runny, more slippery, and this actually helps with moving this secretion, moving the enzymes in the secretion around in the oral cavity, right? So it has more water, it's more liquidy, let's say that way. Whereas the sympathetic system is going to encourage a more viscous, a thicker, more protein-rich saliva. And when we say protein-rich, we don't mean that it has more protein. We mean that it has less water. So the protein is more concentrated, and this secretion is much more viscous. It's not as easy to move around the oral cavity. And this is that dry mouth that we talk about. A lot of medication causes dry mouth, and that's because of the sympathetic nervous system involvement. All right, so let's look at acid and how the stomach secretes acid. Let's look at the biochemical mechanism here. All right, so once again, we have a capillary down here. We have a nice cell. Now, this is a parietal cell. We know parietal cells are responsible for hydrochloric acid secretion. So this is a parietal cell. And we know this is the apical surface. And this is the basolateral surface. Now, unlike absorption, the direction of flow here is not from the lumen. So this is the lumen. It's not from the lumen to the capillary, but the other way around. So hydrochloric acid needs to be secreted onto the surface of the stomach. And so the direction of flow is going to be from the cell and released out into the lumen. Now on the basolateral surface of the cell, we have a protein here. We'll talk about the function of this protein. And then on the apical surface, we got another protein. We'll talk about the function of that protein. Now within the cell, basically we have this reaction that we talked about before, where carbon dioxide plus water under the influence of what enzyme? The enzyme here that influences this reaction into carbonic acid. Carbonic anhydrase. Yep, so carbonic anhydrase is going to bring about carbonic acid. Right? Now this carbonic acid will freely dissociate into protons and bicarbonate. Right? We looked at this similar mechanism before. And now we have free protons that are going to be pumped out of the cell. That's going to be the hydrogen component to our hydrochloric acid. But the way that they're pumped is via an antiporter, right? That secondary active transport that we mentioned earlier. So potassium is moving into the cell. Right, we have hydrogen moving out, potassium moving in, and this is going to hydrolyze ATP to ADP and a phosphate, right? So that's going to harness the energy for the movement of that molecule. All right, so that hydrogen is pumped out, and that's the hydrogen component to our hydrochloric acid, right out onto the lumen. The chloride component comes in because of the exchange of bicarbonate on the basolateral membrane. So on the basolateral membrane, this pump is basically exchanging bicarbonate. Right, so this bicarbonate is going out of the cell, whereas chloride is being pumped in by harnessing that energy. So we've got chloride moving into the cell. And once we have a concentration of chloride building up in the cell, what's going to happen? What's going to happen on the apical surface? So chloride is being moved into the cell against its gradient. The concentration builds up within the cell. It's going to freely move across the apical surface, right? And that's going to be the chloride component for our acid. So together, the hydrogen that's being pumped out, along with the chloride that's freely moving out, is what forms our hydrochloric acid. And this bicarbonate is going to be released down into the blood. Right? So what we'll notice here is that the equal amount of acid that's being released, the equal amount of bicarb that's being released into the blood. So there's that directly proportional relationship between acid secretion and bicarb production. All right? Any other questions here? Mm-hmm.
For the pump on the apical membrane, is that ATP coming in?
That's ATP being hydrolyzed to move.
What's the arrow like?
Oh, sorry, that's potassium. Mm-hmm. All right, any other questions? So potassium is being exchanged for hydrogen on the apical membrane, and bicarb is being exchanged for chloride on the basolateral membrane. All right, and then when that chloride comes in, freely moves across the apical surface, mixes with that hydrogen. We have hydrochloric acid, right? That HCl is hydrochloric acid. Mm-hmm. It is coming in from the blood. Mm-hmm. All right, any questions on this? Now let's. Okay, so let's continue on here. Okay, so that was the mechanism of acid secretion, and we talked about the importance of acid. What was the most important, well not most, but what was one of the important components of, or important functions of acid secretion, the acidic environment in the stomach? Hmm? Yeah, but we talked about one of its functions. It had an effect on a precursor substance, right? Remember, because pepsinogen secreted by the chief cells needs that acidic environment in order to be converted into pepsin, right? The active form. So the acid secretion is going to be influenced by three factors: the parasympathetic nervous system, the presence of gastrin, and the presence of histamine. Now, histamine has a paracrine effect, meaning that it's going to stimulate the cells that are right around the cells in its immediate vicinity to secrete acid. The effect is the same for pepsinogen. So these same three factors have an effect on pepsinogen secretion. Now we want to bear in mind here that pepsin secretion, or pepsinogen, is directly related to hydrochloric acid secretion. Both of these are released in a response to the parasympathetic nervous system, gastrin release, and histamine. Alrighty? So I'll say that again. The presence or absence of the parasympathetic system is going to stimulate the secretion of gastrin. Gastrin is going to stimulate both the production of acid and the activation and release of pepsinogen. All right, let's talk about the regulation here. So the cephalic phase regulation is going to include these factors we talked about: the sight of food, the taste of food, the smell of food, chewing and swallowing will all elicit the parasympathetic nervous system. Once the parasympathetic nervous system is engaged, we're going to influence the release of gastrin. Gastrin is secreted from the stomach. It's also secreted in the intestines and from the pancreas. Once that gastrin is released, what are the two things that are going to happen? We just mentioned them on the previous slide. What are the two things that happen in response to gastrin release? All right, so pepsinogen is one, and what was the second one? Acid, right? Pepsinogen and acid. So guys, think about that in a flow chart sort of way. Sight, smell of food, parasympathetic nervous system, gastrin, both acid and pepsinogen. Right? Think about those chain of events. All right, so let's look at this flow chart here. All right, so this is the same flow chart I was just describing. So the sight and smell of food, chewing food, swallowing food is going to excite the parasympathetic nervous system. That will encourage the G cells of the stomach, there are G cells in the pancreas, as well as the duodenum. They will secrete gastrin. Gastrin will then influence two responses: acid secretion from the parietal cells and pepsinogen secretion from the chief cells. Right? You want to keep that flow chart in mind. The sight and smell of food, the parasympathetic nervous system, gastrin, and then acid from the parietal cells, and pepsinogen from the chief cells. Right? All right, let's look at the gastric phase. So this is down in the stomach. So basically, the presence or absence of food, the presence of protein, the distension of the stomach, liquid in the stomach is going to elicit the gastric phase of regulation. And again, this has to include both the short and long reflexes, So this will trigger gastrin release And we know from the previous slide that gastrin will then trigger acid and pepsinogen release. All right, and moving on to, okay, so let's look at a flow chart that describes that gastric phase. So the gastric phase, once again, we have the presence or the distension of food in the stomach. So once your stomach is full, this response is going to be elicited. The chemoreceptors and mechanoreceptors detect that stretch or the presence of those molecules. It's going to elicit both reflexes, so both the CNS and the enteric nervous system directly, and that's going to stimulate gastrin release from the G cells. Gastrin release should have twofold response. I keep saying that gastrin has a twofold response: acid secretion and pepsinogen secretion from those two different cells. All right, and then let's talk about inhibition of secretion. So can this be inhibited? Can this be stopped, or does this just happen extensively? So the gastric phase is inhibited. We inhibit secretions when food leaves the GI. When food is removed from the stomach, we sort of inhibit this whole feedback, this whole response. Meaning, sorry, meaning we decrease gastrin, we decrease acid, we decrease pepsinogen. So the movement of food from the stomach down to further parts of the GI stimulates this inhibition, which is sort of pulling the brakes on gastrin release. The intestinal phase can be inhibited by a few things, by food moving through the duodenum, by osmolarity, by the presence of fats and acid, as well as by distension. So the stretch will inhibit the intestinal phase.